We begin by explaining to the jury what is DNA. We don't belabor it. Usually the crime labs testified first. But we explain that the human body has like 1 trillion cells in it, and most of them have a nucleus in the center, and in that center of the nucleus is the DNA. And that DNA is 2 complete copies of the human genome, one from your mother, one from your father. Each copy is about 3 billion DNA letters long, divided up into sections called chromosomes of 23 chromosomes. So we have 23 pairs of chromosomes. One of the pair comes from your mother, the other pair comes from your father. And if you were to zoom in on a chromosome and take a look at it, what you'd see is text. In chemistry, it's not like reading a book, but for a cell it is. It's actually one-dimensional text that it can read using different enzymes and proteins. And that text has a 4-letter alphabet of nucleotides abbreviated A, C, G, and T, and that's what we're looking at with DNA. We're looking at that essentially text and the information that it contains. We then explain what a short tandem repeat is, because it's helpful if jurors know what an STR is. On the left is a metaphor of an encyclopedia in 23 volumes, and a particular region, a locus or location, is a paragraph in a volume on a chromosome in that encyclopedia. And for STRs, it's typically 100 to 400 DNA letters long. And the process that it has, what makes it have variation, is how long the molecule is. The whole point of these short tandem repeats, which I first worked with in biomedicine, is that you want to detect variation. If you're looking for differences and everything at that locus is the same, you're not going to find any differences, you're not going to distinguish people, you're not going to identify anybody. But if you have a lot of variation, that means there's a lot of different variants. And at each locus, the system is set up that you can have about 15 different variants on average. Each variant is called an allele. And the way the variant occurs is that you have a stretch of DNA that's fixed, and then a number of repeats, say typically 4 DNA letters long. And so maybe it repeats 10 times, and then you get a constant region again. But the variation is in the number of repeats. In the population, you may see that unit, that word repeated 5 times, or 6 times, or 9 times, or 13 times. And the more times it's repeated, the longer the DNA sentence is. And then the output is to measure how long the DNA is. That's done on a DNA sequencing machine, sometimes called a genetic analyzer. So this was back in the day, the 1990s, when sequencing was very expensive, but measuring the length of DNA fragments was very cheap. So the idea was to measure not the content of the DNA sentence, but its length. Because there was an exact correspondence. If you measure the length, you subtracted off how long the beginning and the end were, and then you looked at what was in the middle, divided by 4. That was how many repeats there are. So that's how complex the math is, just subtracting and dividing by 4. And here's an example of what it looks like with a famous poem, Take Me Out to the Ball Game, that actually has everything in it. The PCR primers are shown in red, Take Me Out to the Ball Game on the other side, at the old ball game. That's how the chemistry from this primer knows to find that paragraph in the entire 3 billion letters of the genome. It just, the DNA finds the DNA, and then it amplifies what's in between. Everything's amplified in between, and in the middle we see, let me root. How many times is the word root repeated here? 10 times. So the allele variant is 10. If it were repeated 8 times, the allele will be 8. Repeated 15 times, it'd be 15. And the more repeats, the longer the molecule. And so that's what a short tandem repeat allele is. And now the key concept in all of this is the genotype. That's what we care about. What is somebody's genetic type? And since you inherit an allele from your mother and an allele from your father at each of these loci, that pair of alleles, that's your genotype. So if you inherited one allele where there were 10 repeats from one parent and 12 repeats from the other parent, your genotype would be a 10-12. That is your genotype at that one locus. That's the goal. The goal is to work out the genotypes so we can compare genotypes for human identity. There are a lot of genotypes that can exist because if you have about 15 alleles, there are about 100 allele pairs. We can't distinguish 10-12 from 12-10. That's why the number's smaller than you might think. And then scientists don't look at one locus. They started originally looking at about 5 loci. Then it moved to 10 about 20 years ago. By 10 years ago, it was standard was 15 loci. Now it's over 20. So if at each location, which is genetically independent of the others, you have 100 possibilities at the first locus, times 100 possibilities at the second locus, times 100, times 100, times 100. You end up with like a trillion possible genotypes. And that's a lot more than the 7 billion people on Earth, so these genetic type barcodes can be compared. That's the concept of genotyping, and for forensics, why you want to make these comparisons. Next, we talk about DNA evidence interpretation. And the idea is to show that the interpretation of simple DNA is conceptually the same as interpreting very complex DNA. Let me show you how. So on the left, we have it's what's represented with one DNA molecule is single source DNA from one person. And imagine it's present in an abundant quantity. There's no limitation. This is easy to genotype. This is like a cheek swab or a pool of blood on the floor. This is easy. This is what they started with 20 years ago, that they hoped all DNA would be like this. It's not. Almost no evidence looks like that now, but that's what they started with. And then the lab, that little arrow that does a lot of work, but it's not a major part of the story today. They do all the heavy lifting, right? They do the extraction and the amplification of the DNA. They take maybe 10 DNA molecules from a swab of a seed, and they turn it into billions of fluorescently labeled DNA fragments of these STRs. And then they detect them by separating them by size, so that the longer fragments you'll see on the right of a picture, like a 12, will be to the right of a 10, which will be to the right of a 5. That's what the DNA sequencers do. They separate by size. So they're very fancy technology. Long talks in it you won't hear from me today about lasers and CCD cameras and separating and machines, robots. It's fantastic. In the end, you get what you see in the middle. It's a cartoon version. That's your data. Is there anything at 5? No. 6? No. 7? No. Something at 10. Peak. Something was detected when the laser shot through those molecules. And there's something at 12. So a very simple DNA like this. The evidence is like a reference sample. It's telling you there's a 10 allele and a 12 allele, and boy, that looks like a 10-12 allele pair. So out of the 100 possible genotypes, this looks like a 10-12. This is the sort of stuff that originally, the FBI hoped all DNA looked like this because it was just so easy to show. And then when you infer to what the genotype is with almost 100% probability, unless there's some issues going on with data, you'd say this is a 10-12 genotype. Note that there's 3 different levels of genotype. There's what's in the biological cell that we can't see. There's the data that we get from the lab that lets us take a peek into what's happening in that small amount of DNA. That's also genotyping data. And then there's the genotype that we infer. When it's simple, there's only one value. And then we can make a comparison to another genotype, develop match statistics, and so on. But what happens when you have 2 or 5 or 10 people in a mixture? TrueAllele's been validated, published in peer-reviewed journals out to 10 unknown contributors. Basically, you practically aren't going to see more than 6 or 7 contributors on even a handgun or a doorknob. So it's, in essence, there's pretty no limit. What if you have a mixture? Now what do you do? Well, how do you know you have a mixture? But the lab does exactly what it does on any sample, whether there's one person in there or 10 people in there. They'll extract, amplify, and separate the DNA and detect it. And now in the center, you see the evidence data. You see these 3 peaks. But there are 3 peaks. So it looks like there's 3 alleles. You only have 2 parents. You're not supposed to have more than 2 alleles for a person. So if you're seeing this at locus after locus on different chromosomes, this is a mixture. There's more than one person in here because there's more than 2 alleles in the data. And they've added their alleles together to give you whatever the pattern is. And so the task is to separate out the genotypes of each of the contributors. Suppose there's 5 contributors to a mixture. The task is to pull out the genotype of each of those 5 contributors at every one of the tested loci. That's your job. The problem is you're usually not going to get a unique answer. It's not going to be 100%. There are some situations with major contributors where it might be 90%, 95%, 80%. But with most mixtures, especially the nasty ones that come back inconclusive, there's a lot going on, and there are a lot of possibilities that have some probability. When you start off, there's 100 possibilities. You can just sort of look at these different allele pairs, and they occur in proportion to how frequently the genotypes are in the human population. Some alleles are more common than others, and so genotypes containing those alleles are more common. The chance of seeing any particular genotype has some probability based on how often it occurs. You may have some alleles that are 20% probability, others are 1%. So there's a starting probability. What we want is for the data to tell us more and move away from that starting population probability. That's what these statistical programs do. They look at the data and they try to separate them. So ideally, what you end up with is not 100 possibilities, each with some probability for one person at one locus, but you get some concentration of probability based on what the data is telling you to maybe 5 or 10 different genotype values. And that change in probability from it could be almost anything to it's more likely this subset, this small group with accurate probabilities is how you get information out of the data. So here on this example on the right, this is just a cartoon example. We're making some assumptions here just for teaching purposes. Imagine we know that the first contributor is the 1012 we saw on the first slide. Imagine that we know, which the TrueAllele computer can figure out just by looking at the data, that this is a 50-50 mixture. It's equal amounts of the 2 people. And now we ask for these 3 most obvious possibilities, what are their probabilities? So why are these the possibilities? Because what's new is that 11. We know the first person the computer's figured out, say, is a 10-12, but now we have to account for the 11. So there's going to be a 10 and 11, an 11 and 11, an 11 and a 12. It's not going to be an 11 and a 15 because there's no 15. I mean, it could be, but that would have very low probability. So it's not in this example. So of those 3 possibilities that are trying to account for the 11, I list them here on the right, 10, 11, 11, 12. They have some probabilities. The reason why one of these possibilities, the 11, 12, may have more probability, say 50%, than the others, is because if you add genotypes together, which is what the computer does, what the computer does is it adds genotypes together that are proposed. It accounts for phenomena like stutter and relative amplification and many other, and distorts the pattern a little bit, and it compares the pattern to the data. So when it's adding together those genotypes, if you've got a 10 and a 12, and you add to it an 11 and a 12, then you'll get 1 dose of 10, 1 dose of 11, 2 doses of 12. And that's what you see in the data. So it's a better explanation of the data imparts more probability. Whereas terrible explanations would have almost 0 probability. You say, oh no, I think this is a 5, 5. Well, there's no peak for 5, and you haven't explained the 11. That would have very low probability. And so in the end, The computer thinks about it, and you end up with, for this one contributor, the second one, separated out from the mixture, a probability distribution for this one of, say, 20 locations. And then when you make a comparison, it turns out that if it is an 11-12, all that's going to happen is instead of having the full match statistic, because it's 50%, not 100%, the number will be reduced by half. So it's almost the same. If you can separate out mixtures up to probability, it's almost the same as regular easy DNA. You just have to adjust by the probability. And everything else is the same in the entire process, the match statistics and everything else. So here is data from one locus called THO1. And I'm going to show you what the data look like. So there are many loci here. I forget which kit this was. We'll see at the end. 15 or 20 different locations. This is one of them. And you see there's more than 2 peaks. We have a 6, a 7, an 8, a 9. 9.3 is sort of special to THO1. That's normal. It's very common. It's a mutation where one of the letters is missing someplace. And what the data look like is horizontally, you're looking at increasing size of the DNA sentences, which corresponds to the boxes on top of the allele. How many repeats do you have? How long is the molecule? Vertically, that's how much fluorescence you're measuring. So that corresponds to more amplified DNA. This is a pattern. What a computer can do is it can use the peak heights. It can work with all the data, certainly the TrueAllele computer. It doesn't need to throw anything out. It can look at the background noise on the bottom. It can look at the medium peaks, the tall peaks. It can look at the pattern of peaks, the highs and the lows. And what actually solves the genotyping problem from the TrueAllele invention is that it can work out from these data and what you're comparing it against as different perfect solutions, what is the variation in the PCR. It actually can go back and tell you how much variation was there in that test tube. And when there's a lot of variation, that means there'll be a lot of genotype possibilities. The match statistics will be low. People won't be as happy if they wanted a higher number. When the variation in the PCR back in the test tube is not as much, patterns will be more definite, fewer genotypes, much tighter, larger match statistics. It's really a direct connection. So the computer, in addition to looking at patterns and peak heights, can also measure the variation in the data, which tells us about the amplification. That's all a bit much. There won't be a quiz on it. That's fine. But what do people do? People are not going to solve a 100-dimensional math problem that can take a computer days to solve. Maybe you have a crystal ball or something, I don't know. But people don't solve these problems. So what the methods from 20 years ago, the way they work is to make it simpler for people by simplifying the data. And that introduces a concept of a threshold. What a lab might have is a threshold, say, just for illustration, say, draw it at 500, labs would usually be lower. And anything under the threshold is thrown out, and the lab doesn't look at it. Anything over the threshold is put on a list, and the peak height isn't used. So a peak height of 500 or 5,000 would be considered the same. By throwing out much of the data, ignoring it, and not using the quantitative information of what's more or less, it's impossible to actually solve the DNA mixture problem. So the human methods don't work except for the simplest cases because it's overly simplified. But the computer can use all the data. And this is how the computer thinks. So we have the same data, and now imagine that we begin just by looking at those blue bars at 7 and 9. So there are 2 rectangles here. The computer says, here's a first contributor. There's an allele pair. And now in orange, there's a second allele pair that's added in. And then a third allele pair in green. It's put on over here. And then a fourth allele pair in purple. The computer proposes patterns like this. What are the genotypes of 4 people at this location? That's what it's trying out. Where do the allele pairs go? more to the left, more to the right. Is there more of them or less of them? That's the heights. And then it can compare the patterns that it constructs, and I'm simplifying here and not showing everything the computer does, with distortions and stuff and such. But it can compare the pattern that it sees of high, medium, low, very high, and medium. When it adds up those genotypes together with the original data, those peak heights, and patterns that are very close to the data, they give more probability to those component genotypes. Most patterns don't look like this at all and are showing you where the probability isn't. And TrueAllele has to think about that as well. When it's all done, for every locus, for every one of the 4 contributors that it considered in this case, you'll get a probability distribution of the different allele pairs. And here we have about a dozen allele pairs that are shown on the bottom. And then we have the probability. So what we see is that the computer looked at the data, and for this contributor to the evidence, this is a 20% contributor. It turns out that the victim was about a 50% contributor, which is why he gave stronger match statistics and larger numbers. This is a 20% contributor. The computer has concentrated probability on about a dozen of these allele pairs. And now we're going to get information. This is all done without looking at the defendant. The computer has not seen the defendant's profile. It has absolutely no idea what you're looking for. It could make a comparison with anybody's genotype. Yours, the judge's, it doesn't really care. It has solved the problem for all trillion genotypes. It now has these probabilities. And all you have to do is decide which of these allele pairs you're going to make a comparison with. You can compare with anyone. And that person will have some genotype. And when we make a comparison, we'll see different ratios. If it's the case, like with the 79.3, that the probability after having seen the data, the evidence probability compared with Brown. Brown is this random probability that we had before we started. So that's the prior population probability. That has some probability for all 100 allele pair values. But the computer inferred something different from the data, and now we're comparing those two. Sometimes what the evidence genotype in blue is greater than the population, and that ratio is the match statistic, like in that case around a factor of 2. Sometimes the brown is greater than the blue. That means the chance of a coincidence is greater than what the evidence is trying to show, and so it's 7/8. If that was somebody you were comparing with, that number would be less than 1, because the blue is half the height of the brown. It would be a half. That would point away from someone. Sometimes the levels are about equal, and then that's not telling you anything. Nothing changed. So what a match statistic is the change in probability at the comparison genotype. Here, Mr. Washington's genotype was 7/9. And when we look at the ratio of the evidence genotype in blue to the coincidence in brown, 27% divided by 13% is around 2. The match statistic for that person, because he has that particular genotype, is 2. That's the first time we make a comparison. So here you have 22 loci. They're listed. The one we just looked at is circled in red. That's the number around 2. And this is the numbers, 1, 2, 3, 4, 5. Each number can produce a value either greater than 1, that might be inclusionary. Some loci are less than 1. With human review methods, anything that doesn't agree with who the suspect is isn't used. That's a little biased, because some of the loci actually do point away, and you want to consider all of them. The numbers are all multiplied together because they're independent genetic locations, and now we can answer the question, to what extent is the suspect in the evidence or not? And we can make the statement that a match between the right rear seat and Dante Washington is 60,000 times more probable than a coincidental match relative to an African American population. We can make the same statements for Caucasian or Hispanic, the different populations have different heights of their brown bars. The different well, what is random? And the way that it's been done around the world is random is done based on ethnic population. So there's the random African American population, the random Caucasian population. Crime labs and the FBI and other groups develop those. So we report 3 match statistics. You can make the whole thing race-free by just using the smallest one. Say it's around 12,000 times more probable. But this is very simple language because we've reduced it to separating out one genotype. We're not making a complex statement about explaining all the mixture data. This is a genotype that was separated from the mixture, so we can compare this one genotype, the 20% genotype, with Dante, Washington, and say, using the smallest number, that a match between the seat and Mr. Washington is about 12,000 times more probable than coincidence. And all those words are understandable to a jury because they know what probable means. They know it's the probability of a match compared to the probability of a coincidence and so on. But it's pretty straightforward. It's unfamiliar. People don't usually think in terms of likelihood ratios, but there you have it. I won't dwell on this, but one of the powers of TrueAllele and why we can report results on very low match statistics. Some people would say low is less than subjective, is less than 1 million. Some people think low is a match statistic under 10,000. But we have reported match statistics of 25. And you may ask, what does that mean? And the answer is we can also compute an error rate. So if I tell you that the match statistic is 25, but 1 in 100 people who didn't leave their DNA would match that strongly, it's not the strongest evidence in the world. On the other hand, if I tell you the match statistic is 1 million and that the error rate is 1 in 1 billion, so the chance that somebody who didn't leave their DNA would have a match statistic at least as big as that 1 million for the defendant is 1 in 1 billion, that's much smaller. So this gives us a sense of how frequently the match statistic would occur for this genotype. In this case, for a match statistic of around 10 or 20,000 that we reported for Mr. Washington, the error rate was around 1 in 500,000, 1 over 500,000. So that gives some frequency context. Okay. What it lets you do is to get to the error rate that extensive validation studies can only approximate, because here we're reporting the exact error rate on this evidence item for this match. It's very specific. And these methods are used in different forms, but the exact approach that we take that considers every possible genotype with great precision is something that's in TrueAllele. And now we can also show with a match statistic that in the same mixture, the victim is present with a match statistic in the quadrillions. So now you have a mixture that's putting the defendant and the victim, is the cab, on the same mixture in his car, the same item. And then we tabulate the match statistics. Now, most of the match statistics to the victim, except one, were known to the crime lab. So the new information that we found was the match statistics to the defendant on the door handle, on the carts, on the seat, and also showing that there was a mixture that contained both of them. So that's the evidence that we were talking about in this case. And that's all we do. We take data from results that labs may have called inconclusive or unknowable, undecipherable, or whatever, and then scientifically put exact numbers to them of whatever they might be. Inclusionary, exclusionary, big numbers, small numbers, whatever it is, having separated out the mixtures into genotypes. And this is showing the powers of 10, which we sometimes need to get into on cross-exam. So a number like 10,000 is a 1 followed by 4 zeros. That's where the number 4 is coming from. These numbers are in the tens of thousands. Because in a lot of the material we provide, we summarize all the computer runs we did, we replicate everything, and then questions will come up Why are you using these numbers, not those numbers? Why did you use 4.2, not 4.7, and so on? So if the jury doesn't know about that, that's kind of hard. But scientists talk in terms of how big numbers are based on how many zeros they have. Okay. The cross-examination started off in a totally bizarre way, where the defender simply focused on race. Nobody had ever seen that before. He was basically accusing me or the prosecutor of being racist. It's like, this is like totally weird. And that's what he dwelt on for a while. He also introduced some bad ways of understanding match statistics that were incorrect, and we discussed that. And so they went back and forth. There was a direct, there was a cross, there was a redirect, there was a recross. And first assistant DA Martin Wade, anybody know Martin Wade? Really sharp, and also I thought he was a bit of a gambler to try to do this on redirect. You know, most prosecutors would just done it on redirect and not go for the fifth round. But what he did is he wanted to undermine the concept that anybody's a racist. And that's because things are reported with different racial groups, which is standard. So there wasn't any merit to what was being said. And so he asked me about any pro bono work that Cybergenetics had done. He asked, have we done any exonerations? And we had just worked in this Indiana case, pro bono, demonstrating that Darryl Pinkins, a man who had been in jail for almost 25 years, could not possibly have been involved in this crime. And ultimately the DA said, I agree with the science. We're not continuing with this. And then his co-defendant, who'd been released earlier, Roosevelt Glenn, had just been exonerated as well. And that was a case that we worked on with the Indiana Wrongful Conviction Group, completely pro bono. It was a fascinating case. We've written about it, spoken about it. I won't go into it. And then we had also worked on this case where some other software was used with this New York case against defendant Nick Hillary. And I also wrote about this one. But it was clear that the operator of the software could adjust some parameters, could change thresholds. And what we showed was that with that software, changing the threshold from just a low level of 30 to 50 would change an exclusion to a number like 10 million. And the judge ended up saying, no, that's not good, reliable science. And so these were successful cases. And then Martin asked me, were these men black? I said, yes. And he ended. That was it. It's like, so it didn't make much of an impact on the jury to be called names when your actual work doesn't have racial bias. The opposing expert went so off the deep end that I wrote an article about what he did. I didn't mention his name. That's not nice. And I spoke about it at a conference. And so this is a paper, a little magazine article for lawyers. Good answers to bad questions about DNA match statistics. As you just saw, what is a DNA match statistic? It's a ratio of two numbers. A probability after you've seen evidence divided by a probability before you've seen evidence. That's what a likelihood ratio. It isn't anything else, and that's what they've always been for decades. And so they make up stories. They tell the jury, well, what if it's not a ratio? Basically, they go sideways. Let's look at the highest probability. Well, that's not relevant. You have to make a comparison where the defendant is or the victim is, not anywhere you want to go. That doesn't make any sense. What if you added up all the other probabilities? Well, that doesn't make any sense. You have to look at where the person you're comparing is. And then they look at just the numerator. The idea is that the match probability of the numerator may be small, but if the coincidence probability is much, much smaller, then a small number divided by a very small number is a big number. And so by ignoring what's relevant, that was the expert. And it was like, why? I mean, why would you distract a jury with stuff that makes no mathematical sense? I won't answer that, but I'm sure you've seen it. So in the end, the defendant was found guilty I don't know to what extent the DNA evidence played a role, but it was evidence used at the trial.